Thank you for your interest in our video. Today we want to introduce the thyroid cancer and its signaling pathways, diagnostics markers and targeted therapies to give you an idea that how important it is to optimize early diagnosis and develop targeted drugs. 1. Thyroid cancer overview. Thyroid cancer has become the most common cancer among all the malignancies. For unclear reasons, thyroid cancers occur about 3 times more often in women than in men. And it is commonly diagnosed at a younger age than most other adult cancers. Several factors can affect a person's chance of getting thyroid cancer. Thyroid cancer occurs more often in women than in men. Radiation exposure is a proven risk factor for thyroid cancer. Sources of such radiation include certain medical treatments and radiation fallout from power plant accidents or nuclear weapons. Genetic syndromes that increase the risk of thyroid cancer include familial medullary thyroid cancer and multiple endocrine neoplasia. Follicular thyroid cancers are more common in areas of the world where people's diets are low in iodine. A diet low in iodine may also increase the risk of papillary cancer if the person also is exposed to radioactivity. 2. Main signaling pathways in thyroid cancer. Thyroid cancer has become the most common cancer among all the malignancies. Several treatment modalities including surgical resection, radioactive iodide therapy, and hormone suppressive therapy could result in good prognosis in most patients with differentiated thyroid cancer. However, such conventional treatment methods are not effective in treating patients with medullary thyroid cancer or anaplastic thyroid cancer. Although conventional treatment methods are not effective for such patients, researches in molecular biology could bring new chances. At present, the main molecular signaling pathways of thyroid cancer including Src signaling pathway, Rtk signaling pathway, NF-kappa B signaling pathway, TSHR signaling pathway. Any changes in these signaling pathways could be used as biomarkers to help diagnosis and predict the prognosis of thyroid cancer. In addition, potential treatment targets could also be found in these pathways. Src family kinase, SFK, is a family of non-receptor tyrosine kinases. SFK could be involved in cell proliferation, growth, motility, migration, angiogenesis, and intracellular transport. Both the direct and indirect activations of SFKs are associated with the progression and metastasis of malignancies. Activated Src pathway could consequently activate several other pathways including mitogen-activated protein kinase, phosphoinositide 3 kinase, FAK, and STAT pathways. These activations finally affect the growth, invasion, and metastasis of thyroid cancer. Potential drug targets and diagnostic biomarkers in this signaling pathway include Src, FAK, MMP2, MMP9, etc., and Creative Biolabs provides high-quality antibodies to these targets. Rtk signaling pathway integrates both intracellular and extracellular signals, and serves as a central regulator of basic biological processes of cells, such as metabolism, growth, proliferation, survival and angiogenesis. MOP pathway could transduce extracellular signals into the cells and nuclei to induce biological responses including cell proliferation, differentiation, and apoptosis. Overactivation of MOP pathway could upregulate the expression of numerous oncoproteins. PX/ACT pathway is a very important intracellular signaling pathway for mammals, which is closely associated with cell proliferation, transformation, metabolism, motility, and development and progression of tumors. The association between NF-kappa B and development of tumor mainly relies on the fact that NF-kappa B could inhibit cell apoptosis. NF-kappa B could influence cell apoptosis by regulating cytokines. It could also inhibit apoptosis by inducing or upregulating the expression of anti-apoptotic genes such as BCL2, or by inducing the expression of TNF-alpha receptor family, cellular inhibitor of apoptosis proteins, and zinc finger protein A20. Specifically, for thyroid cancer, NF-kappa B activation was found in PTCs, FTCs, and ATCs, and researchers suggested that activation of NF-kappa B could promote the differentiation of PTCs and FTCs and thus play important roles in each stage of thyroid cancer. We provide various antibodies IKK alpha, IKK beta, I-kappa B, P50, REL and other main targets in NF-kappa B signaling pathway of thyroid cancer to help our clients research and meet their goals. TSHR could combine with TSH and thus stimulate the growth of thyrocytes directly or indirectly by stimulating growth factor. 2 intracellular signaling pathways could be activated after the combination of TSHR with TSH. Each signaling molecule in these pathways could interact with the signaling molecules of other pathways including WNT, VEC, and MOP pathways to form a network. Although TSHR's genetic and epigenetic alterations do not directly lead to carcinogenesis, it has a crucial role in tumor growth, which is initiated by several oncogenes. With years of experience, our scientists can offer high-quality antibodies targeting TSHR signaling pathway in thyroid cancer to meet our clients' demands precisely. The table lists targets on the main signaling pathways in thyroid cancer, and our scientists can provide you with far more than a list of target antibodies. 3. Molecular detection markers for thyroid cancer. Thyroid cancer is the most common endocrine malignancy with steadily increasing incidence over the past few decades. Although standard strategies for the management of thyroid cancer offer optimal outcomes in thyroid cancer patients with favorable histological types at early stage, challenges arising from diagnosis and therapy still exist during clinical practice. A number of genetic alterations have been described in thyroid cancer, which provides an unprecedented opportunity for the identification of novel diagnostic and prognostic molecular markers as well as novel therapeutic targets. The table shows molecular markers that could be used as candidate biomarkers for thyroid cancer. 4. Targeted therapy for thyroid cancer. For the past several decades, thyroid cancer has been the most common endocrine tumor, with a 5% increase in incidence each year in the USA. Recently, a number of scientific advances have illuminated some of the molecular pathways responsible for thyroid cancer. This growing knowledge raises the hope that it will soon be possible to develop specific therapeutics tailored to these molecular changes. Targeted agents, listed in orange boxes, in the picture include those in clinical use, colored in red, and those in preclinical or early phase development, colored in green, for the treatment of thyroid cancer. As the preferred supplier for global customers, Creative Biolabs are confident to provide you with first-class services covering a full range of applications. If you have any need for antibodies, please contact us. We will be in 100% of the effort and offer 100 points of the products.